Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we would like to take a moment to thank our ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Please mark your calendars for our next Dr. GPCR symposium held on Friday, September 22nd on the topic of GPCRs as therapeutic targets. The symposium is free, but you must be a Dr. GPCR Ecosystem member, which is also free. Please see the menu button for the symposia page at the top of our welcome page to check out the program and register as a presenter. You don't have to register as an attendee. You just have to be part of the Dr. GPCR Ecosystem to get access to the links to join us. We also have set aside 2 hours for networking and poster presentation on Kumo Space that day. The posters don't have to be fully formatted. Just come with a few slides and have a chat with your community members about your favorite GPCR or other signaling proteins that you're working on. If you'd like to present during your work during the networking time on September 22nd, please complete the submission form available on our website. There is no limit on the number of posters we can accommodate. To present, you'll only need to provide us with the title of your presentation, a 1-minute video abstract, and come with a few slides to help you discuss your work with your colleagues. Everyone is welcome. If you have any questions or comments, or if you'd like to present your work but don't know how to join us, please feel free to email us at hello@drgpcr.com. What are you doing between November 2nd and 4th? Join me and the Dr. GPCR team at the 22nd edition of the Great Lakes GPCR Retreat at Chateau Montebello in Quebec. We at Dr. GPCR are proud to be supporting this iconic meeting. I've had a blast help organizing this meeting this year with my colleagues from the University of Ottawa, the University of Montreal, and the University of Sherbrooke. Visit gpcrretreat.org for more details on the program. The early bird registration and poster submission deadline is September 8th, and the late registration deadline is September 21st. Hurry up and join us between November 2nd and 4th. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm very excited to have with me very recently PhD minted Dr. Ilana Kotliar. Ilana, welcome. It's nice to have you.
Thank you, Yamina. It's so great to be here.
Thank you so much. And thank you again for inviting me to your thesis defense on Zoom. It was really, really impressive.
It was really wonderful to be able to share it. And I think doing it hybrid is really the best of both worlds.
I thought so too, but you could feel, I could feel that I wanted to come and congratulate you, and that's when the Zoom stopped. I was like, I know hybrid is good, Zoom is good, but in person is always better.
Agreed.
All right, so let's start at the beginning. Please introduce yourself and tell us who you are and what do you do.
So my name is Ilana Kotliar, now Dr. Ilana Kotliar. And I am a postdoctoral associate in the lab of Dr. Thomas Sakmar at the Rockefeller University in New York. And Dr. Sakmar's lab is also where I just completed my PhD training as part of the tri-institutional program in chemical biology. And so that's a little bit about my immediate background. And of course, we study GPCRs. And my thesis research was really had two different focuses. The first was on method and tool development, and the second was on studying protein-protein interactions between GPCRs and a small family of accessory proteins called RAMPs, or receptor activity modifying proteins.
Awesome. The audience may or may not know this when they're going to listen to it, is that you and I have Rockefeller University and the Sakmar lab in common. I don't think we overlapped though.
No, we didn't.
But we did meet at a GPCR retreat, and we've been in touch since then. How did you get to pick your lab when you picked your PhD lab?
So actually, I think this is an interesting story where I think the moral is, when you go into grad school, or sometimes when you go into whatever your next step is, you think you want one thing and you wind up with something that's even better than you could have imagined. So when I came into TPCV, as we call the program for short, I had some ideas in mind about who I wanted to rotate with, and I had my first and my third rotation set, but I needed that second rotation in the middle. And my program does this really great thing where different PIs come in and give lunch talks during your first summer. So you get an exposure to all the different types of research that you could potentially partake in. And Tom gave a really nice talk. And after that, I went and I met with him, and he was fantastic. And the project that he suggested also seemed super interesting. And so I rotated in the lab. And I guess the rest was history. But in the moment, it was not such a straightforward decision. My first year advisor, Dr. Tarun Kapoor, who was also one of the co-chairs of the program, he said that you need to look at three things. The first is the PI, the second is the science that you're doing, and the third are your colleagues, and in that order of priority, because the PI is the one thing that will certainly not change, although the type of experiments you do may evolve, and certainly from the start of your PhD to the end, the people in the lab will likely also turn over. And so I kind of use that framework as my guide and kind of evaluated my three rotations through that prism. And I felt that Tom was really supportive PI, both in terms of my science and my professional development. I loved the topic, and I was doing some microscopy and fluorescence imaging, and I really liked that. And then also the people were fantastic. So that's how I joined the SACMAR lab.
I think it's super valuable what you just said because one, I'm glad that you had a good experience. Two, I think for all future graduate students who are thinking about picking a lab, and what you just mentioned can very much be applied to picking your postdoc or picking your job afterwards, no matter what that is you have to really keep in mind that you really need to find a supportive PI, a supportive boss. You need to be doing science that's really interesting to you. But at the same time, you have to have great colleagues, even though in an academic lab that the turnaround can be quite high depending on who's graduating. And sometimes you have these labs where everybody starts at the same time and all of a sudden everybody graduated.
Right. And there's no gradual transition. It's just like flipping a switch.
Exactly. And that's very valuable. I always say, and I realize that I didn't come up with this. I just heard it somewhere and I keep using it as, finding your next position or your next job. It's kind of like dating. You really want to make sure that you like the other party.
Right.
Because if you don't, it's going to be a very long date.
Yeah, you want to make sure it's a good fit. Like there's no good or bad. It's just, is it good for you?
Yes, absolutely. What got you interested in science in general?
So I think I was always interested in science, and I know this will probably sound very cliche, but from a young age, when I was very little, I wanted to be an astronomer and study space. Uh, and I had great plans, and I studied the solar system, and I was very interested in that. But then once I got a little bit older, I was very lucky to have some really fantastic science teachers in grade school. And one in particular, Susan Katoff, she was my chemistry teacher my freshman year of high school, and really kind of opened my eyes to how cool chemistry was, and kind of sparked in me wanting to really understand concepts and understand how things work at the most basic level. And so that kind of set me on the course of chemistry. I was also always interested in biology. And so then when I got to college, I went to Cornell and I majored in chemistry and chemical biology there. I chose to focus on the chemistry side because the classes really let me build a conceptual understanding from the ground up. But at the same time, I wanted to see how all this could be applied to understand how life works and hopefully to improve it. So my research interests were always on the biochemistry side. And I did a lot of different research experiences during my 4 years of undergrad to kind of figure out what were the maybes or the yeses that I enjoyed, and what were things that I didn't want to do, because I kind of always knew that I would want to go to grad school and get my PhD.
You mentioned something very interesting about, getting those experiences and figure out what are the yeses and definitely what are the nos. And that's also very important to figure out what you don't want.
Yeah, I think when we're doing experiments, we're always going after the positive result, right?
Yeah,
But
yeah.
when we're having our life experience, I think it's important to also factor in the negative. So one example for me is that I was interested in metabolism research. And so at one point I tried to do the training to learn how to work with mice. And I realized like I just couldn't do it. And so that was a very clear no for me. And so that helped me to define my path a little bit more. So it was a positive experience, even though in some ways it was a very negative result.
No, I agree with you, and I can't do that either. I think it's just I don't have the personality to work with mice, definitely not
Very tough.
with any animals at all. That's a very tough, and it's very important to know where your limits are and in every aspect.
Yes.
So you mentioned how you told us how you picked Tom's lab.
Mhm.
When did you ever first hear about GPCRs? I'm just curious if you remember the exact moment or not. There's no wrong answer here.
I for sure learned a little bit about GPCRs in like basic biochemistry coursework at Cornell, but I don't think I really appreciated how complex and important they are until my rotation. And then because so actually I shouldn't say that I learned in biochemistry, because in my undergraduate research in Dr. Henning Lin's lab, I studied a GTPase. So I actually that was probably my first introduction to GPCRs in that context. But since I wasn't studying them directly, that was more like a peripheral piece of information. And then only when I got to the lab and then I really started to understand that on and off switch is just kind of the first level of many, many, many more levels.
No, I think the reason I asked this from everybody, and sometimes we have guests who say, well, as you just mentioned, it was just in the periphery. I kind of stumbled on it, or I ended up working with a lab and we discovered something and we said, this could be really interesting. Or others would say, it was fascinating from this class and I couldn't just stop thinking about GPCRs. You mentioned that your PhD project, well, your PhD work had two major streams.
That's
Since it's fresh in your mind, can you tell us a little bit more about at a high level, what were the objectives of your project? What did you add to the field in general, and how people can appreciate or can use from a methodology perspective the tools that you built?
Sure. Okay. So hopefully I'll hit all three of those points and I'll talk about each of the streams separately. So, and I will say that both of these were very collaborative between myself and Tom and some great scientists in the lab of Dr. Jochen Schwenk at SciLife Lab in Sweden, where I also had the privilege to go to a few times and work with them and do the experiments there. Okay, so on the methods and tool development, we applied an assay. It's called the suspension beat array, or SBA, which is an immunoassay. And you can think of it as a very fancy multiplex co-IP that's read out in a flow cytometer instead of with an immunoblot. And so this technique has been used to profile plasma, but we wanted to apply it to study GPCRs. And this work was actually started by my predecessor in the lab, Dr. Emily Lorenzen, who started this collaboration and put in a lot of the foundational work and did a lot of the proof of concept, hard experiments. So we wanted to use this platform for developing or validating, I should say, antibodies against GPCRs. And on the other hand, the second part is to study GPCR protein-protein interactions. So on the first part for validating anti-GPCR antibodies, it was really cool to be able to use the SBA for this because we can test a lot of different GPCRs in parallel. So you can figure out not only if an anti-GPCR antibody is binding its intended target, but if it's cross-reacting with probable off-targets, which is unfortunately a problem for anti-GPCR antibodies since GPCRs share pretty high homology, and for other reasons, that generating anti-GPCR, specifically antibodies, is challenging. And so we did this and we used antibodies from the Human Protein Atlas. We tested more than 400 of them, and we wound up validating 248 of these, which correspond to 154 different GPCRs. And this was a paper that we recently published, and we also created this very cool interactive web interface to accompany that. Since most people study one GPCR very in depth, so they can hopefully use this as a tool to look at what we ate in their research. Now, on the second half of my work, which is on GPCR ramp interactions, GPCR ramp interactions have been very well studied for secretin family GPCRs and ramps, and the Sackmar lab actually has some longstanding interest in secretin family GPCRs, but there's a lot of other non-secretin GPCRs, and those interactions with the ramps have not been studied very much or at all. And so what we wanted to do is map the GPCR ramp interactome for receptors across all subfamilies on the phylogenetic tree with the 3 ramps. And so we again used this suspension beta array, this SBA immunoassay to do that. And these are results that are currently being written up, but we do see some widespread GPCR ramp interactions, which was super exciting. And then you can say, so what? They interact, or you have some evidence from the screen that they interact. If we want to hedge our bets a little bit, what does that mean for GPCR biology? And so we picked one specific GPCR ramp pair, that of an itch receptor called MRGPRX4, and we characterized its interaction with the ramps and saw some interesting effects of interaction with ramp 2 specifically, which maybe down the line we can leverage for anti-itch therapeutics in the context of cholestatic itch.
Wow. That is awesome. And you made a great point when you mentioned the difficulty of having good anti-GPCR antibodies that are specifically recognizing the right receptor. And there's also this issue with antibodies in general, but specifically against GPCRs for application. So you validate it in an application, so in a flow-based application, these antibodies. And I love the fact that you have this interactive tool. We'll definitely post the link onto your podcast episode page if people want to take a look at it, because it's absolutely worth checking it out. I remember seeing a talk from Tom where he gave the link, but I don't know if I saved it. So now I will definitely ask you for it so that we can.
Yes, I will send it to you. I also have a QR code, but probably a link might be better.
Yeah, I think that might be useful. So that's really great. I asked this from all our guests, but having worked on validating all of these antibodies and worked on specific, ramps with characterizing with this itch receptor, do you have favorite GPCR?
This is a tough question because most of my work was studying 215 of them. So I feel like I almost can't pick favorites, but if I had to, I would say it would be between the itch receptor that I just mentioned, MRGPRX4, and probably the calcitonin receptor-like receptor, or CalCRL, which is our prototypical ramp interacting GPCR. It was the one that led to the discovery of the ramps, and it's also a really good example of how GPCR ramp interactions are therapeutically relevant because of the anti-migraine therapeutics, and there's now a lot of them that target the CalCRL complex with ramp 1 for migraine. So I love to use Calcarel as an example. And of course, we also use it as a control in a lot of our assays. So it would be between those two.
Awesome. And it doesn't have to be a GPCR. We had guests who said, oh, actually, I have favorite G proteins, or beta-arrestins are my favorites. Or I think actually, Andrew Tobin's favorite was a phosphorylation site, which
Wow.
I thought was very, very creative.
Then I would probably pick ramp 2.
That's what I was getting to. I wanted to know if there's a ramp preference there. So
Ramp
you're
2
definitely coupled to ramp 2.
Yep. It's different from the other ones.
Yeah. I was wondering if you could tell us a little bit more about, life as a graduate student, considering, it's 2023. COVID is not such a long, hasn't it's very close to us. Although I think we don't hear COVID news anymore, but it's still among us since the vaccines have been deployed and a lot of people get vaccinated. But how was it for you to do your PhD during such a tough period?
So I think on in different dimensions, it affected me and other graduate students differently. So since I was in my second year when the lockdown happened, I probably was luckier in terms of the timing, where the lockdown meant I had a few months right before my candidacy exam to prepare my candidacy exam. So in that way, I think I got relatively lucky with the timing. But coming back to the lab and coming back on campus, I think myself and other students, since there was no more social events or very limited social interaction, it kind of created a little bit of a siloed experience. And now coming out of that, I think the friendships that I made in graduate school before and or with people within the same year or similar years as I were, those kind of stayed. And there's kind of like a gap in terms of who you know of your colleagues who are of younger years in their graduate school journey. So I think that missing social component really affected all of us and to different extents, because of course, as you pointed out, we tried to do things over Zoom, but it's really not the same as being in person, really forming those bonds, but also just having those casual conversations about your science, what experiment you're doing, and kind of maybe picking up some new connection or getting some insight that just otherwise wouldn't have happened. So that's definitely a downside. I think the introduction of Zoom in general was in some ways positive because we can have things like this and you can have meetings across the ocean, and it kind of normalized that. So it is for sure a silver lining, even though nothing replaces being in person. Maybe we now have a newfound appreciation for that. The other side was in the lab and the shortages of the supplies was very frustrating sometimes when you just want to do the experiment and you want to get to the next step, but you don't have the flasks or something like this. And I think that actually hasn't fully went away. So that's probably one of the very negative effects that we're still seeing today as like a long-term consequence on the research environment. But I think this will also take some more time before it kind of re-equilibrates.
I think so too. And I remember seeing even very recently, a couple of months ago, actually, messages on Twitter, people asking for plates or a specific catalog number saying, do you have 10 plates? As soon as our order comes in, we'll happy to replace those. Can you please help us out?
Right,
And
right.
it's just
Tissue culture flasks, those were in hot
wow.
demand, not available.
Wow. So you thought twice about passaging, like changing your flask. Oh,
Yeah. Yeah,
wow.
we'd actually like calculated out how many more passages and all these things that you would otherwise maybe take for granted.
Wow. Yeah, it's I think it was a very demanding 2 years.
Yeah, I think also one other thing that was very specific to me is that since my collaborators are in Sweden and the experiments that I needed to do would be in SciLife Lab there, and all the borders were closed, it was very stressful to figure out first how to ship all these biological samples from New York to Sweden, and second, how I could go myself. So that was probably the biggest overall challenge of my PhD.
How did you solve it? Or I mean, the shipment is one thing, but shipping you.
We were checking the renewed guidelines basically every week, and as soon as they opened up, I bought my ticket. But in the background, we were also planning alternative like visa permissions, all these sorts of administrative things that you don't have to do during normal circumstances. So we also got quite lucky that when we really needed it, they finally opened the border, and we found a great courier service for the samples. So eventually it worked out very well.
Oh, I'm glad. Yeah, it sounds like a hassle. I mean, I didn't travel during these restrictive periods, but just the thought of, well, if it's closed, the border is closed, it's closed. But even once it opens, thinking about the risk, yes, you're traveling with a mask. It's not fun to have a mask on for hours on end. And then the shortage in tests and then having to do a PCR test. Otherwise, it had to be negative in order to let you on the plane. Just a lot of
The test,
hassle.
yeah, the hour window before the testing. And this was before you had these like at-home tests.
Yeah.
So going there, it was stressful but okay. But then coming back, getting like the test done there to come back, it just adds a lot of logistics, which
Yeah.
are of course surmountable, but it becomes a bigger and bigger question that you have to optimize for.
Oh, agreed. And also, running experiments in a new lab in a different country where you don't have the comfort of being in your own lab, basically. And it's already tough, but adding COVID restrictions, it just adds a little another extra, stressful um layer
Yeah,
to it.
it was different there because Sweden has very different kind of rules and regulations. So in some ways it actually felt a little bit more normal.
Okay. Aren't they, wasn't it Sweden that decided not to have restrictions at all? Or was it the Netherlands?
Yeah,
There
they
was a
were in the
country.
news a lot. Yeah.
Yeah. Wow. And I remember watching the news and we were like, okay, I think, you know, it's a decision that had its consequences as well on that end.
Yeah, it's also just a very different like number of people, size
Yeah.
of country.
Yeah.
So it's hard to compare.
Yeah. Definitely. Definitely. All right. Top three aha moments that you had that shaped your trajectory as a scientist. It can be any type of aha moments, but anything that happened, whether from an experimental or an experience standpoint, where you're like, okay, now that kind of shaped where you are today.
Um, I feel like a lot of aha moments are yet to come since I'm like relatively early on in my career, but let me pick out a few and hopefully they'll probably be from all over, but hopefully something will be interesting to whoever is listening. So I think one aha moment I had that is probably very actionable for others also is how important it is to be organized, and that planning what you do and keeping careful records and having things laid out in such a way that you can then go back and understand what you did and somebody else can also is really important. It's not enough to just work or do experiments and generate all this data. It really is important to take the time and plan out what you're going to do and do what maybe would be considered a less interesting experiment first, but to systematically build up to what your ultimate end result is. Um, so I think that's one big one, especially the part about really methodically planning your experiments. I think I learned that from a scientist in the lab, Dr. Emily Serrato, and she does these really complicated 384 well assays. And working with her on this MRGPRX4 itch receptor story really gave me insight into that. So I think that's maybe not so much like a glamorous aha moment, but I think it's very important for success in the lab, and I think success in general professionally. So that would be one. I think the second aha moment, and I had this in undergraduate, is that what your main focus is not enough. So like your studies is not enough, and in research as a student, your bench work is not enough. You have to do something outside of that and develop skills and work with people for other interests. So, for example, by giving presentations or by getting involved in outreach, if that's something that interests you. Because you need this perspective both personally to kind of inform you to figure out what kind of things you like to do, but also to develop skills that you won't learn when you're standing at the bench on your own. And I saw this a lot at Cornell also with all the different clubs. And I think I transferred that over to my time at Rockefeller, and it was really instrumental to my happiness and also to my figuring out what I wanted to do post grad school. I think those are two of my probably biggest insights. I'm trying to think of a third one to round it out.
While you think about it, I have a couple of questions because I like how you approached answering this question. It kind of blends in with my follow-up question about advice to junior scientists who want to contribute to the field. I think it's a very important, as you said, to have the ability to do other things that are not related to your work and not spend all your time at the bench, because you need to have that perspective. You need to set that aside, let your brain in the background do whatever it needs to be doing so that you can come back fresh and rested. What are some of the things that you like to do when not at the bench that gave you this opportunity to, freshen up and do something else that you feel like really helped you get back to the lab on Mondays?
So I did different things over the different years. One of the main ones was being involved with my program. So I served on the student organizing committee, and we helped organize recruitment events. And that really, I really loved doing that because I felt like when I was the one being recruited for different programs and coming to interviews with big eyes, looking at all the beautiful labs and impressive PIs with their research and trying to parse where was the best fit for me, the students I talked to really were honest and helpful and willing to spend their time. And so I was really joyful to be able to pay that forward and talk to students who I was in their shoes a year ago or 2 years ago, etc. So, and that also helped me kind of see who was incoming and pay it forward, but also was joy for myself as well. And also to see all the great things that were happening in the program, because our chair, Dr. Derek Tan, he's always trying to make it better. So it's also always great to see how we were changing and improving. So that was one of the main ones. And I guess the second extracurricular that helped me, I guess, to always come to lab energized was my outreach work that I did. I worked with some other fantastic students in the Tri-Eye Outreach Committee, or TORC as we called ourselves. And in 2020, when fresh from lockdown, we realized that kids who were applying to grad school, who were in college, now everything was remote, they didn't have this easy access to mentors the way that we did when we were applying to grad school. And so we started this program that we called TIMI, the Tri-Eye Mentorship Initiative, which was a fully virtual one-on-one mentorship program where if you were applying to grad school, you fill out a short application. And we recruited mentors from the Tri-Eye community, and then we matched the mentors with the mentees based on interests. And then they would have their one-on-one meetings and communications, and the grad students would help the applicants with writing their personal statement, figuring out where even to apply, what to expect from interviews, and these sorts of things. And the program wound up being very successful, and it's continuing this year, even though I've graduated now under new leadership, which is really exciting to see something you start continue. But that was also extremely rewarding for similar reasons.
I love it. And I think while you were mentioning these two examples, I was thinking of transferable skills. I hate the term, but doing these extracurricular activities allowed you to acquire or to perfect your communication skills, your people skills. You got to work with different people. You got to figure out how to adapt to everybody's style. And at the same time, you learn how to really put yourself out there. And I think it requires, at least for me, it does require extra energy when I go to meetings and you have to chit chat with everybody. It's very energizing, but at the end of the day, I'm done. At the end of a meeting, I just don't want to talk for a week because it was just so draining. And having these regular opportunities to talk to different people allows you to learn to socialize might not be the right term, but it's important to be able to talk to people at different levels and helping
and have constructive conversations.
Absolutely. And helping releases oxytocin. So that's also kind of the reward, and releases oxytocin on both ends.
Yes.
So I think that's really great. You also mentioned learning how to, and the importance of being organized, and the importance of taking notes, and not only doing experiments just for the sake of doing experiments. Do you have any good or bad story that taught you these skills, or that, made you realize how important these skills are?
Do I have any? I don't have any huge, like, spectacular, good or bad, but I think it's one of those things that I picked up from other students I saw and I interacted with, but I only realized how useful it was when it was time to prepare the manuscript or put together the presentation. And if I hadn't done it, it would have, at that point, it's too late. You can't go back and figure it out. If it's there, it's there. And if it's not, and you're not sure, you have to redo your work. Yeah. So I, in some ways, prevented that, thankfully. And I've tried to teach it to others that I've had the opportunity to mentor, that I give them my system. Like, this is how I number my experiments. This is how I keep track of them. This files, this program, etc. And you try it, do what works for you, but make sure you have a system so that in a month from now, when we ask, well, why did we choose to include this condition? We have an answer. And that's definitely happened countless times, even though I don't have a specific one top of mind.
That's very valuable, and because it's important, and it is more important than you think.
Maybe one bigger scale example is for this big GPCR ramp interaction screen that I did with my collaborators. And this was like almost 2,000 samples in an assay. And we had a very definite amount of time to do it because the flight here is on this date, the flight back is on this date, and you have to make it work. So we met, I think, almost every week for like a few months leading up to it, working on the spreadsheet, organizing the plate, organizing the reagents, organizing the samples, blocking off the time to make sure everything is timed accordingly, booking the instruments, etc. And then when we got there, even though we did have to make changes, that was only possible because we already had such a strong framework in place, because you always will have to make some sort of changes on the spot. And then the experiments worked and we were able to do all of them. So I guess that's probably my best example of how it really pays off to be organized and plan things thoroughly.
I think so too. And I think it's very important. And this is a translatable skill to everything, not only working in the lab, to anything and everything that you do. Anything that is, oh yeah, actually everything. I'm gonna stop talking because the word everything says it all. It's important to be organized. It's important to plan things out. It's important to think about your next steps. It's important to think about how you think you're gonna get there. And it's important to have a plan for it. And so my next and final question, and if you're comfortable sharing, what's next? You're currently a postdoc in Tom's lab. You just finished your PhD not too long ago. What next? And if you're happy to share what next, tell us a little bit why the next step the way it
Sure.
is.
Yes. So I'm now postdoc, like you said, in Tom's lab. And after this, I have accepted a position as a life science consultant at the firm EY Parthenon, where I will continue to be based out of New York, which I'm very excited for, in addition to all the things that the job entails. But when I do share what my next steps are, people are often curious why. And I think I gravitated towards the job that I chose, I guess, for a few reasons. The first is I really wanted to see how science works in the business world and learn kind of the other half of the coin and see how great ideas, therapeutics, ways to help people, how that actually comes to market and makes an impact more in real time, as opposed to working on something that maybe in 10 or 20 years get translated. So I kind of wanted to be in that exciting space. And consulting is a great way to learn a lot of that very quickly. So that was one main reason. And the second is I realized I really liked the planning, the presenting, the working with my team, Tia, Annika, Leo, Johan in Sweden, and Tom, and also Marcus in Sweden. And so I wanted to pick something that matched the parts of the job, the parts of grad school that I liked for my job. And in consulting, it's a lot of analysis, teamwork, presentations, meeting and talking to key opinion leaders and other really interesting people. And so I also try to optimize for that.
I think that's very important as well to note that you need to figure out, not just you, but everybody needs to figure out what they like. And it goes back to what you said, that very quickly you were able to figure out what are the yeses and what are the nos. And
Right.
you focused on the yeses and chose a path forward that has, for a majority of the time, the yeses, the things that you actually like to do, because then
Yeah.
it's a pleasure to do them and it's not like going
I think
backwards to work.
I think this can tie into my third insight, which is how important it is to talk to people. And I mean, leverage connections. It sounds very like formal, but it's important to ask people for help or for mentorship or for guidance, because that was really instrumental to me many times throughout grad school, before that as well, in getting here, and after or during, when I was looking towards the next steps, talking to people who had made similar decisions a year before and could tell me their rationale, and so I could start to parse it. So the human element and how important it is, I think, is probably like the third and biggest insight.
I could have not said it better. Thank you. I think I'm not gonna even comment. I think this was you just said it so well, and it is very important. Ilana, thank you so much. Good
Thank you so
luck,
much for having me. Thank
of
you.
course. And then we're gonna stop recording and let people wonder what we talk about after we stop recording. But thank you again for joining me on the podcast. I really enjoyed talking to you.
Thank you for all the great work that you do and for the Dr. GPCR ecosystem.
Thank you. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our Dr. GPCR podcast guest and our team members, Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. Please mark your calendars for our upcoming 2023 Dr. GPCR Symposia. Don't forget to visit us and check out the program. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem. Join us today. Also, please subscribe to the Dr. GPCR newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. With any questions or suggestions, please email us at hello@drgpcr.com. Until next time, stay safe.